I want to talk about this data mainly, and we have heard already from Peter how data are managed in EBiSC. And this is a basic process here, just as an overview on the left side. So if a cell line is going to be banked in EBiSC, it goes first through a registration process where available data on the line are registered. Then these data will be validated, exported to a central data information management system, and finally displayed in a catalog for e-commerce. And along this way, several players are involved. First, EBI with their tools and services, and of course, the bank itself, which provides additional data and validates the data experimentally, which are deposited in the registry originally, and the data will be completed if there is something missing. So I want to touch on these issues today. One is data governance, then what data standards and what the validation process means. I want to go a little bit more deeper into the ethics and informed consent issues, and finally talk about a little bit about tools and interoperability. The next. Just use the computer. So what kind of data are we talking about? And when we look at the process for iPS generation, we have first the donor, then we have the reprogramming steps, the cultivation step of the iPS cells, and then there's also the differentiation potency, but also there are cells derived from these pluripotent stem cells which are being used and applied. And all these steps, all these points create different types of data. If you want to now enter these data for pluripotent stem cell lines, you can go to this registry and decide whether you want to register a cell line and its data for the pluripotent stem cell registry, or for EBiSC or any other specified entity. We have on the bottom here the types of data are kind of structured, which is general information, so who deposits the data, which institution do the data come from, the donor information, the ethics data available for that line, derivation data, culture conditions, characterization, meaning is the line pluripotent, what kind of markers are expressed, are they contaminated with microorganisms, is there genotyping information available, and was a genetic modification done with this line? The first step, actually, which we do and we have talked about, is to give this cell line a name, which serves as a reference or a standard, a unique identifier for that line. And this naming is associated immediately with the provision of a biosamples ID, and Peter has mentioned that, which then allows to link these names or cell lines with other information available together with the biosamples ID. Then the actual data will be registered, and we have defined some minimal criteria which we use as criteria to say, well, this is a pluripotent stem cell line according to the data we have provided, and the ethics information is reasonably good to say it can be used in EBiSC. These minimal data will be validated, and other data, of course, can also be added, which are not part of the minimum criteria. And I will tell you later in a minute what these minimum criteria are. Of course, there are other certain data which are in the information management system which are not in the registry. For example, disease annotations or certain LIMS data which have been generated during the banking process are not fed back to the registry. These are batch-specific data which we do not collect in the moment. So what kind of data are registered? And I already said there are data related to the generator, so general information, contact information, owner, who's the owner of the line, who's the distributor of the line, who generated the line, and donor data. And the most important here, which we consider a minimal criterion, is consent. We need information that the consent is available for a certain line. We need also a unique identifier, so a name basically. We need this contact information. All the other data we also collect or are able to collect, like phenotype of the donor, genotype, family history, clinical records, but they are not part of this minimal information criterion we need. Then we collect also source cell data or primary cell data, like for the fibroblast, a cord blood cell, something else, and data on microbiology of the donor and of the primary cell. And then the pluripotent stem cell data themselves. So the derivation data, part of the minimal criterion are gain, potency, and identity. So we need to know how we can identify, authenticate these cell line with SGR or SNP data, and what is the potency? And this potency, we have two criterion. One is expression of markers, which are typical for pluripotent stem cells, and the others is actually differentiation potency. I go back to the diversity of assays for that a little bit later, and what kind of problems that causes. Here also data on cultivation, characterization, QC, omics data, genome sequencing data. This all belongs here. Also the application grade. So is this a GMP line or a non-GMP line, or clinical grade line in a way, can be entered here. And finally, we want to have some feedback from the generators or from users of the line, like what kind of projects these lines are being used in. For example, what kind of clinical trials are being done with certain cell lines in the future. Are there publications of the lines? Can we get feedback from users? What is the legal framework in different countries for using of these lines? And are there new standards or new legal frameworks available? So this is all information we can use. Minimal information is potency, identity, unique identifier, and consent, plus some contact information on about the person who registered the line. So how we validate these data, right? We need to validate these minimal information data, and then once it's validated, you get this released button being in green, and then everybody can see the line. And this is, in the moment, a manual process and only a plausibility check. So there's some risks that the data are not correct, not perfect, not correct, and some risks of confidence. The confidence would be increased if the data are verified by an independent lab. And the question we have here that is an issue of course of feedback. Right? We have for some data we have EBiSC, which feds back data which have been generated through the banking process, and that adds of course confidence. But there are also data which are not we cannot check whether they are right. The only data we really check are the ethics data, the consent information. Also helping in rising confidence are assays which are standardized assays. So here you can compare with existing data and have a much better plausibility of assays, for example, pluripotency, to say, well, we trust these data. How we can increase confidence, of course, as I said, feedback from users can increase confidence in data, or when we get cells from a qualified bank or qualified lab. That gives us much better confidence that the data are right and okay. We ask for binding statements by the providers of data. So the provider has to confirm at the end that he or she puts the data in to the best of her or his knowledge. We issue certificates, and we can only issue certificates when we have a certain level of confidence that the line is what it says it is. And these certificates are being used by the European Commission to basically confirm their usage in European funded projects. We also have a network of national representatives, and then when critical issues arise, we contact them and ask them, well, for example, is this person who enters a line a real person? Or is it a computer bug, something like that, which didn't happen until now. So here, just in a nutshell, the validation process again. We have data from hundreds of depositors and many, many lines. And to have a manual validation process is eventually not feasible anymore. So we need some kind of automation integrated here. It's also very expensive to do it manually. So for the certification, we need this validation certainly. Only lines that have been some level of confidence can be certified. We need interaction and feedback from users and depositors. But we also need to automate this process, some programs to validate the data against pre-existing data or data which come in later on. And as much as possible, we want to use standards, the data standards or ontologies to basically navigate with the data and make this process, the validation process, more automated. And one issue here is standard assays. So if people use 10 different assays to measure pluripotency, it's very difficult to automate this validation. I want to talk a little bit now about ethics validation process and how we codify ethics to make that possible. And this is one thing where we try to introduce some kind of automation and comparability. If we get an ethics consent information sheet, which we require for each line, it's in Chinese, it can be in Spanish. German is okay for us, but other languages are difficult, and we cannot read everything. So we ask the users to fill out some easy questions, like has consent been obtained from the donor of the tissue from which iPSCs have been derived? So is there any consent? If this is a no, we can already dismiss the line and we cannot validate it, and so on. And we have several fields with similar type of questions which we want to fill in. For example, permitted users. Is it only for research? Is it also for other applications? Is it for a certain project, or is it open to other projects? Is there commercial exploitation necessary? How is the storage and access to material regulated? Can people access, can everybody access the lines, or is it a national access consent? Is access provided to the source cell line or not, and so on? Are there any benefits promised to the donor of the cells or not? Can the donor withdraw from her or his consent? Which has all some implications. And this scheme was actually one of the first steps being done in the EBiSC project. Other points are medical access to medical records. Is it there? Whom to contact to get this? If yes, was there an ethics committee approval for making these iPS cell lines or donation of the original tissues? Are there any third-party obligations or restrictions? Are there any other constraints to the usage of the lines? Are there MTAs available? And then a big part is data and information. So how is that to be handled? And we had quite some struggle with entering genetic data. This is complicated because we know now that genetic data can be used to re-identify a person. So even if it's open access by consent, there might be some issues, but if it's not open access, which is very often the case, in the consent information, we need to ask certain questions like, is a collection of genetic information allowed? And is the storage of genetic information allowed? Does consent prevent the dissemination of genetic information? And has a donor been informed that their donated biosamples or cells may be tested for the presence of microbiological agents or pathogens? And how is that fed back, perhaps, if something is found like HIV, to the donor? Has the donor consented to receive information discovered during the use of donated material? And all these issues are all kind of codified in the system. So the first issue we also encountered is how to describe genetic modifications. So and this is a very interesting cell line, usually with genetic modifications or gene-edited lines. So how to enter these data? And there's one format, which is the HGVS format, which is kind of a standard. It's shown here how it works, and basically, so this is a sequence ID, and if you want to use this standard, many people don't know actually how to put a genetic data in this standard into a database. So it's difficult to read, and it's also difficult actually to compare lines based on this HGVS format. So one solution would be to use this kind of approach to enter 25 base pairs on each side of this single base pair modification of the gene, and then you can just perform a simple BLAST search with all the lines in the database and find the lines with a certain mutation. Another tool has been presented by Peter, who can do a allelic mutation screen of the whole genome sequences if they are available. So this is an issue we still think is not perfectly solved, how to enter genetic information and how to search for genetic information. And another hurdle comes with the regulatory environment surrounding genetic information. And this will even become more severe in Europe with the implementation of the European data regulation, where privacy-related data are very restricted. So a typical question for data access for genetic data is shown here. We want a cell line, this genotype, E2, E3, E4, in the ApoE gene. And in the EBiSC, as Peter mentioned, this is no problem because these are open. They're all open access. But if this is not available, like for many informed consent information forms, this is not open access, then we have a problem to find these data, and we need to go through a data access committee. And there might be different data access committees. EBiSC has one, but EBiSC, for example, has a different one, and other providers might have the data deposited in different databases or with a different data access committee. So what we can do in the moment, and this is this Beacon project from the Global Alliance for Genes and Health, which facilitates an interface to genotype queries, but does not return information that could be construed as violating anyone's privacy. So basically, this is a question: is there a line with a certain mutation on chromosome 3? Yes, but we cannot tell you which line it is. This is very unsatisfactory. You can only get this information once you have approval from a data access committee. So I will not further discuss that. Only say that this issue will certainly increase when we get more and more reporter cell lines with genetic modification. isogenic line, other gene-edited lines, or lines with cultivation-based modifications. And this will create a whole kind of genome vault surrounding iPS cell lines, which eventually will only be manageable with machine learning and artificial intelligence tools to select lines, to check alleles, to define pluripotency, to draw conclusions from these genetic information on the phenotypes of the lines and on the potency of the lines, or on the phenotypes of derived lines, on cell transitions, and the expected changes when genes are modified. So this is just a visionary thing, and I'm sure we will go into this direction. More and more genetic information or whole genome information is available. So another issue I want to just briefly mention is the cell types, cells derived from iPS cell lines. So there are several cell types, there are also organoids, and the question is, do we also want to have a registry for these lines? What kind of information should be there? We have developed for a few years ago a tool called Cell Finder, where we can basically register all cells of the human body, and the mouse too actually, and then we can basically zoom into this human body down to the cell level and look for gene expression patterns in this line. There are several tools. Several tools can be associated with that, saying, well, this line, this cell type looks different from the cell type I have derived from my iPS cell lines, which should also be a cardiomyocyte, but it looks like a little bit different, or is a certain cell type in my human body. To structure that, there's an effort to standardize that. There are also ongoing efforts. I want just to mention one, which is the MIACAM, the minimal information about cellular assays for regenerative medicine. And this considers data on projects, on the source cell, on assays being used, on experimental technology being used to characterize cells, on the data themselves, and then, of course, on the cell production and stem cell QC, and this will be further developed into a MIACAM 3 tool for cells, basically every cell of the human body. And most of you probably know that there is now a huge initiative ongoing to create a human cell atlas, and this falls very well into this area of research. So what are the challenges and future work that I see should be done? And the first thing I would like to say is that we need to consolidate and enforce the ethics codification to make it more screenable. What kind of information is there? What has the patient, the donor, consented to use the cells? And there are now several consent information forms available, derived, for example, established by the iPSC or by other bodies, which cover almost all of the possibilities. So, and it's some kind of modular system where you can take out things or add other things which you want to have in this consent information. So codification based on such a modular system should be really quite straightforward. But what I also want to mention is that we should enforce the use of standards, which are defined by the community, which makes it much easier to register the data, which is a very elaborate process in the moment, but also easier to validate the data. So if you don't know which pluripotency assay to be used, and I appreciate that this is a moving field, we all don't know that yet, we will have always a lot of assays. And each assay has some pluses and some minuses, and but it's in the end very difficult to validate. So we also should, I think about developing a genetic environment or vault for iPS data and relevant access tools to these genomic data. And one tool has been presented by Peter, one possible tool. There are others also available. New interface features and tools should be developed we use as much as possible ontologies for searching and connecting to other data areas which also use ontologies. Images have been mentioned, so we are not very good in the moment with collecting, searching, and collating images for cells and for assays. And what about iPS-derived cells? What about the differentiation potential of iPS cells and the usage of the iPS cells? We should be able to get this feedback. Enable feedback mechanisms, and maybe we could also distribute the validation process more into the communities and do it manually within on a single site. This is a part of all these points are part of interoperability. So if you want to link external resources like EBI or NCBI to iPS cell data, we need interoperability. And one point is a reference name, which I already mentioned, but others are standards, which allow automated systems, standardized data archives and banks. And with that, I would like to finish. Thank you very much.
Yeah, thank you very much, Andreas. Questions? I would start first with a question. Maybe I didn't got it, but if you want to refer to a data entry to a specific cell line from a publication, how would you cite it in the materials and methods? You have a kind of digital object identifier, or how would you directly cite it, or with a unique identifier?
Well, I would cite it with a standard name, which we generate, okay, plus the biosamples ID, okay, which is automatically associated with that name. And the biosamples ID allows you to enter information which is available in other databases like EBI or NCBI or RIKEN in Japan, which all use these biosample IDs. We also provide a QR code to this name, link to this name.
Yeah.
So on your ethics tick the box thing, you said has a patient given permission for use of the tissue, but you didn't say explicitly for iPS generation, because often sharing the line means that it has to be not just donation of waste tissue, but specifically for iPS. And so the wording is perhaps not satisfactory for sharing the lines in many cases.
Well, in some cases, it's not acceptable. So it's not a must criterion that the donor has said, yes, it can be used for iPS generation. It's not a must criterion to validate the line or to register it. But for some people, it is an issue. So there need to be an explicit yes towards that iPS generation.
Hi, could you tell us a bit more about the Beacons project? How does it define violation of the patient's privacy?
The Beacons, the basic principle, and maybe Peter can say more about that, is that you shouldn't give out data which can re-identify a donor, genetic data. So at least not openly. So the Beacon project just searches a database and tells you, well, we have that particular line with that particular mutation, but we cannot tell you which it is. We cannot even give you let's say 100 lines, and one among these is the line you're looking for. We can only ask you to apply to the data access committee. And if the data access committee finds that you are a real researcher and your project is ethically all right, then you can get the information about this line. But that makes it very difficult, for example, to screen for cohorts. If you have 10 lines, each line is from a different, let's say, you need a different data access committee. It's already a huge barrier for many people to go there, and that might be one reason why people rather make their own lines.
More questions? If not, I would like to have a final question as well. One comment first, of course, this would be perfect when it would be linked to a physical specimen biobank, of course, where you have every cell as a master cell bank. I mean, this would be a wish, I think. I think you agree. The second thing is the use of the cells is not only limited by the informed consent, it may be limited by IP and ownership. Is this something planned for the future to incorporate this, or is this something which is on purpose not?
Well, we asked for ownership information, and we also asked for, I haven't detailed that, but we asked for IP restrictions and restrictions on usage in general.
Okay, thanks very much.